Welcome to Engaging Ideas, where we bring information and ideas from the Academy to the community to help think about real-world matters. Our topic today is diabetes, a complex and serious disease affecting an estimated 30 million Americans and hundreds of millions worldwide. But rather than describing the implications of the disease, as significant as they are, we're going to look inside human cells at molecular processes that may hold the potential for new approaches to the treatment of diabetes. While it is a complex subject from a scientific point of view, our interview looks at the topic in non-technical terms. Joining me to lead this intracellular journey is Dr. Cedric Asensio, Assistant Professor of Biology at the University of Denver. His laboratory at DU focuses on understanding the internal functioning of cells in relation to complex diseases such as diabetes. Over the past 3 years, his lab has been awarded over $2.5 million in research grants from the National Institute of Health, the American Diabetes Association, and the American Heart Association. Dr. Asensio holds his BS and PhD degrees from the University of Geneva in Switzerland, and prior to joining DU, he did his postdoctoral training at the University of California, San Francisco. He's the author of over a dozen publications and an expert on the topic. So let's begin. Professor Cedric Asensio, welcome to Engaging Ideas.
Hi, Jim. Pleasure to be here.
So for those of us who are not scientists engaged in biological research, let's start with the basics. What is diabetes and what causes it?
So diabetes is a disease that occurs when you have too much glucose in your blood. Another way to say that is you have too much sugar in your blood.
Okay.
So this glucose comes from the food you eat, and it's going to be used by your cells as a source of energy. So we need glucose for your body, your brain to function. But too much glucose can be toxic, and in particular, having too much glucose in your blood for too long can lead to severe health problems. Now, so to prevent accumulation of glucose in the blood, our body uses a hormone called insulin. And the job of insulin is to help your cells uptake the glucose and use this glucose as a source of energy. In doing so, it's clearing the glucose from the blood into the cells. Now, in some people, sometimes you don't have enough insulin or any insulin at all, or your body doesn't use insulin very well. Mm-hmm. So in that case, the cells can't uptake the glucose, and you have this buildup of glucose in your blood, which leads to complications. Now, to go back to your second part of the question as to what causes diabetes, right. It's a complex multifactorial disease. By that I means there's not one culprit. It's the combination of your genes, the environment in which you evolve, and your lifestyle. But in all cases, the main underlying problem is a defect in insulin secretion and or a defect in insulin action. That leads to a state of hyperglycemia, too much sugar in your blood.
So insulin is the key here. Let's just take that a bit further. Just what is insulin and how does it relate to diabetes more specifically?
So insulin is a hormone. It's a protein hormone, so it's being secreted by specialized cells in your body, and it goes in the blood, so it circulates in the blood. It can act on many different tissues and organs. The main targets of insulin are your muscle cells, your fat cells, and your liver. And if we wanted to summarize kind of the overall action of insulin, the job of insulin is really to normalize your glycemia, so to reduce the amount of sugar you have in your blood.
Okay.
But here's where it gets tricky, is that too much glucose is toxic, but too much insulin can be a problem as well, because you can fall into a state of hypoglycemia where you don't have enough glucose and you can't get your brain going because you don't have energy reaching your brain here. So the system has to be very finely tuned to work well.
So you talk about fine tuning. That's an interesting description. Can you elaborate on just what fine tuning the system involves? What's involved in that?
Yes, definitely. So this fine tuning means that insulin needs to be released quickly on demand.
Okay.
So if you think about it, one of the best way to accomplish this is to have a reserve pool of insulin pre-made that is ready to go. And that's exactly what happens. So insulin is being made and stored by specialized cells. They're called beta cells, and they're found in your pancreas. Now these cells, in addition to making and storing insulin, they also have a special property whereby they can sense and monitor the changes in blood glucose, and in response trigger the secretion or prevent the secretion of insulin.
So you mentioned the secretion process. How does that work?
So this process is called regulated secretion, and I think if we want to understand this process properly, we have to take a look at the basic organization of a cell.
So I believe you have some drawings that'll help us do that. So perhaps you could bring us through the first drawing.
So if we take a look at the basic organization of a cell, you'll see that your cells have compartments that are separated from the rest of the space in the cell by a membrane. So it's a physical barrier. Mm-hmm. It's kind of like drywall in a house to divide the space into rooms. Now, having compartments enable to create a specific individual environment here. So now you can optimize certain functions or certain reactions. If we go back to the house analogy, you have a kitchen. That's a room with a specific function. You can prepare food. So you've optimized the environment to prepare food. Mm-hmm. And you have a bedroom, a bathroom. So it's a similar idea within your cells.
So let's now link that back to insulin. How do these compartments in the cell relate to the formation, storage, and release of insulin?
The relationship with insulin is, I mentioned that insulin is being stored within the cell. So it's actually stored within one of these compartments.
Okay.
This compartment has a name. It's called an insulin granule. It has a lot of insulin inside, and you can have many of these granules within one cell. Now, the life of these granules begin at the Golgi apparatus. The name is not very important. It's another compartment. Okay. So that's where the granules form. And actually, this process is not very well understood at the molecular level. And this is why this is a key focus of our research in our lab here at DU. So you're forming this granule from this internal compartment. From there, your granule is going to move from the middle of the cell towards the periphery of the cell. And these granules will accumulate there. So you have a pool of granules that are ready to go. The next step is these cells will sense the change in blood glucose. So you have this increase in blood glucose. And as a response, now the granules will move even closer to the membrane of the cell and physically fuse with it. And in doing so, they're actually releasing the content, so all the insulin can be released outside the cell. And now your insulin can end up in your bloodstream and do its job. So this process is called regulated secretion, and it's a complicated process. It's regulated by many different proteins within the cell. But if we summarize, it's really the cell senses the change in blood glucose, as a response moves the granule, fuses the granule, and release just enough insulin to normalize your glycemia.
It really shows the complexity of the human organism, and in this case at a molecular level.
Definitely.
Yeah, just amazing. So the work that your lab is doing, and others around the country and around the world, is fascinating. But I guess the question is, where does it lead? We're doing this kind of basic research, but what are the potential implications of the work you're doing in other labs on medical practice and in people's lives?
That's an excellent and important question. So what is the role of basic research in 2018?
Right.
So the point of basic research is to generate fundamental knowledge about the world, okay? To create new ideas, to challenge dogma, to come with new theories and principles. But why should we care?
Right.
So if we take an example and we think about basic biomedical research, which is what we do. Mm-hmm. If you imagine the future of medicine, you would want physicians to be able to offer personalized treatments to their patients, as opposed to like one-size-fits-all kind of approach.
Right.
So there's actually a term for this that is being used in the media, kind of a buzzword. It's called precision medicine. So what precision medicine is just giving the right treatment to the right person at the right time.
Okay. Okay.
And if you think about it, physicians have been doing this for quite a long time, or they're trying to do this. If you need a blood transplant, for example, to save your life, you're not going to get blood from a random average person. You're going to get blood that matches or is compatible with your blood type. Mm-hmm. So we can do this because we have enough knowledge of the underlying biology of the process. So this is a relatively simple scenario. Okay. But in the near future, you would want physicians to be able to offer this type of personalized medicine, precision medicine, for any type of diseases, including diabetes, where you have a lot of contribution from your genes, your lifestyles, and your environment. So we'll only be able to achieve this if we have a deep and complete understanding of all the underlying biological processes and mechanisms that are associated with the disease. This knowledge will come from thousands of labs around the world asking and solving basic research questions day after day. So maybe I can give you one specific example from the kind of research we're doing in the lab.
That'd be great.
So recently we've identified how a particular protein called HID1, the name is not important, influence how much protein hormone can be packaged into one of these granule as you're forming the granule. Okay? So this is a basic research question. It's been done here at DU with undergrad and grad students. Now, somewhere else in the world, there is a team of physicians, and they have identified patients with very severe neuroendocrinological defects. So they don't know why these people are sick. It's a new syndrome. What they know is that they found a very specific change, a mutation, in the protein that we're studying in the lab. So our work doing basic research now can address, we can reproduce this mutation in the lab and see whether the function of this protein is affected and maybe give an explanation to why these patients are sick. So we're not curing the disease, but we're providing maybe an explanation for why they're sick. And in the future, we can hope that more basic research like this can act as a stepping stone for future development of therapeutic drugs.
It's a perfect example of the value of basic research and the way it migrates into real-world solutions to very difficult and important problems.
Definitely.
Well, Dr. Cedric Asensio, thank you so much for sharing this intracellular journey, the story about insulin and diabetes, and the possible implications of your research on medical practice and indeed on people's lives. I hope you'll join us in the future as we bring more engaging ideas from the Academy to the community. I'm Jim Grismer. Thank you for watching.